Hello. So today's topic that we are going to discuss is a new genre of analysis. It's all about studying the DNA and protein at single cell level. You can do DNA alone, you can do protein alone, or you can do combine both of them. Within DNA, you have the option to look at the mutations or SNVs. It's mostly done with the Mission Bio, a company that is specialized on these. So let's go through this. The company specifically advertises that single cell genomics and DNA, where you can look at the SNV or single nucleotide variation, copy number variation, and DNA plus protein. The platform name is Tapestri platform. You can come to this website and find details about it. Now, for example, you take a set of cells. It could be any sort of cancer cells or healthy cells, and from there the single cells you isolated and you run through this Tapestri platform. We assume that we will be studying the DNA and protein, both of them in combination. So this series of tutorial within this will be going through, elaborate on a step by step, specifically on the interest of the bioinformatics pipeline development, optimization, and as well as functional, based on our biological interest, to analyze this DNA plus protein single cells multi-omics data. So they have an algorithm that's called Mosaic. This website is specifically regularly updated on the purpose of this analysis. You see, recently they have updated to the version 3.0.1, and they have certain instructions on how to run DNA, CNV, protein, signals, copy number, variation, subclone, the algorithms, as well as different types of plot, heat map, line plot, bar plot, multi-heat map, tree graph, fish plot, a lot of them. And they have some multi-sample DNA plus protein. Either way, so as I will be going through step by step of this data analysis, I'll show you a complete tutorial with explanation of each steps on how to perform the bioinformatics part and optimize it in a perfect sense to find our biological interpretations or what we are looking from the biological perspective. So first, I think you can look at one of our video where we specifically I have shown that how to create a specific anaconda microenvironment that serves the interest of installing this Jupyter notebook, where created at the very first in the Linux system. In short, you can go through the video. Created a new virtual environment. Within that, created the new Anaconda environment, and within that Anaconda environment, after its activation, created a specific port of Anaconda that functionally will be only opening this specific environment. So you can see out of many, this is one of them. Let's write it as a IBM TNA protein tutorial. So let's assume that you have, what you have to do actually, let me go through. For example, you run it within the Tapestri with the latest version of the flow cells they have, or chemistry specifically. And when you run it, after that you upload your first Q files. They have specific cloud environment where you just need to upload it. I can show actually separately different times. I will show it definitely. I just forgot. Let me write it down actually. I'll show it. So, and then you will get the .h5 files. So now you want to load those files. So they have certain specific, the mission buyer has created this specific, you can go to the starting points in here with this. So the package you need to install in here. So if I just, for example, Kanda, please, you can just run it and you will see. So we have this, let me just make it bigger, you can understand better. So this is the mission buyer package that's installed in here. So this is something that we will be using for analysis. So let's start. This is a short video at the beginning point. So I will show you how to import the package of mission buyer and then just load the files, as simple as it. So let's import mission buyer, import mission buyer.mosaic.io as Mio. This was as simple as it. So let's import the packages. So as we have this, now let's load our files. Take our, for example, this is our H585s. So we put it like, so what do you find in here? Now the next fuzz in here is that this is the path we have shown. Now we want to load our H585. So we go to the sample, for example, we defined the Mosaic, mission by Mosaic, I as Mio. So we write Mio, we load it. Our file is in H5, path is H5 path raw. So here comes the question, what is this means? So the first thing, do you want everything to be, because this is a filtered file, we don't want everything to be loaded in here. So we run it like false. We don't want all barcode to be loaded, all raw counts to be loaded. So we set the false, which exclude those things, and what's filtered will be loaded in here. So next fuzz is we go for the filter variants. So we go for filter underscore variants. Just keep it careful in here, version to version they change it. So apply filter was previous version, the current version has this filter variants, and future version could be different. So I'm just giving a heads up. So this we set as true. What does it mean? It means that filter variants which has been filtered in the DNA variants will be only loaded. So it's very critical and very important as well. So if you just load all of these and your variant of interest is not in there, then you need to change it at false, so that will be loaded. Otherwise only the filtered will be loaded. So that can be done in different way, and it depends on you how you want to actually lead with this. That's very interesting part. So next part will be like, that's a single the true. So we set up single equal to true. So usually we set single as a true because each sample is unique and it's important to keep that track in there. And another parameter is that some people add the whitelist. So it's like it's a condition where they set it like okay, if the filtering even exists, that still we want our specific variants to be present in there. So to do so, you can specifically define, okay, I want this filter to be added. Like you can add it like whitelist, but it's not necessary. It's not always necessary. Most of the time we define it later on. You load all of those and then you define, okay, now I don't want them. So that could be another thing. So I would suggest, like, think about it. It's nothing like an obligation or anything, but you can always run with this. So either way, that's all right. So for example, I want this. This is the list of the variants that I want. So I add it up like this. Now what we had? We have these. So we need to end it with now you want our sample. So you can see this is loading our samples. So the samples are loaded. And now if you require, like, I want to see some more information. Now if you want, like, okay, I want a little bit more information from here. So we will go through the extensive analysis from the basic filtering to everything. You can write something like that. Okay, DNA, want the heat map of the DNA. So we set up like attribute well, these are the different chromosomal positions. So we'll come through this one by one. We'll divide the clones and everything. So this is the basic. Just to look at the basics in here. You loaded your samples, so that's the basic. So we'll go through a lot of these things one by one. And if something is coming like, okay, you are not finding what chromosome X, so let's remove that. And let's rerun. Hold on. Restart and run on. There must be some mistake somewhere. Okay, see. Yes, like, that's mistake. It looks fine now. And you can come in here and make it. So this is the basics. That's how the way we will, if you want DNA and protein to analyze the data, and I will go through like every detail of how to select the CNV, SNV, and everything that's you require to be an expert on DNA plus protein data analysis in terms of identifying mutation, making all sort of graph that includes the protein alone with SNV, with going through the each of the subclones, every single details you will require. That's the purpose. Okay, thank you.